Welcome back to the new video. In this video, we are going to focus on a new topic called as immunoglobulins. Immunoglobulins are also called as antibodies. They are they are glycoprotein molecules which are produced from the plasma cells in response to any antigen or immunogen. So, immunogen is a type of antigen that has the ability to trigger the immune response or immune system in the body. Here, plasma cells means plasma cells are the specialized white blood cells which play a very crucial or important role in immune system. They basically produce or originate from the B lymphocytes and are responsible for producing and secreting the antibodies, that is immunoglobulins, to help in fighting against the infections. They play a very important role in the immune system by recognizing and binding to the specific antigen or a pathogen and helping the WBC cells to neutralize those antigens or pathogens or microorganisms in by killing or eliminating the harmful pathogens from the body. Structurally, they are Y-shaped in configuration. So the immunoglobulins are Y-shaped configuration. So let us start the video with different types and more about the immunoglobulins. So in this slide, we can see the basic structure of immunoglobulins, or you can say antibodies. So basic structure usually it contains four polypeptide chains. In among these four polypeptide chains, two are called as heavy chains and two are called as light chains. So here, this portion, that is the lengthy portion, this portion is called as heavy chain. That is called as heavy chain, and the small portion is called as light chain. Light chain. And these chains, that is these heavy chains and the light chains, both are linked together by a bond called as disulfide bond. Here we can see the bond. There is a bonding between the chains, that is they are joined or bonded to each other by a bond called as disulfide bond. Then another portion is constant region and variable region. So here variable region is used to bind for the specific antigen and constant region is used to determine the antibody function. Means it is the determining factor of the antibody function. So in this slide, types of immunoglobulins. So totally there are 5 types of immunoglobulins. Here we can see the types of immunoglobulins. The classification is mainly based on the type of heavy chain they contain, and there are totally 5 major classes of immunoglobulins. In that, first one is IgG. It is the most abundant antibody which is available in the blood, and it is responsible for long-term immunity and secondary immune response. So IgG is responsible for providing long-term immunity, and it is it's responsible for producing the secondary immune response. And the next one is IgA antibodies. So it is found in mucosal areas like respiratory and digestive tract, and it gives protection against the pathogens in these areas, that is in the mucosal areas. And the next one is IgM antibody. IgM antibody, it is the first antibody which is produced during any immune response. Means whenever there is an infection occurs to the blood, the first antibody which is get activated is IgM. It is mainly found in blood. And the next one is IgE antibodies. So IgE antibodies are responsible for producing allergic reactions, and it is providing defense and protection against the parasitic infections. The next one is IgD antibodies. So IgD antibodies are very important and in play a very important role in initiating B cell activation. B cell activation. So here the B cells contain some antigen recognition receptors, that is called as B cell receptors. So these B cells contain some B cell receptors on their surface, which is called as BCRS, which is called as BCRS, that is B cell receptors, and they are helpful in recognizing specific type of antigens. So these are the five main class of antibodies, that is IgG, IgA, IgM, IgE and IgD. Discuss these antibodies one by one. The first one is IgG antibodies. IgG means immunoglobulin G. So it is the most abundant antibody in the human serum. So accounting about for approximately 75% of the total immunoglobulins. So total compared to other antibodies, it is present in excessive range, and it plays a very important role here in neutralizing the pathogens by following mechanism. That is, it facilitates the phagocytosis mechanism, and it is also helps in neutralizing pathogens by activating the complement system. So complement system we will see in the next slide. What is complement system? This slide we are going to discuss regarding the subclasses of IgG antibodies. So immunoglobulin G, that is IgG, has some subclasses, namely Ig1 is there, then Ig2 is there, Ig3 is there, then Ig4. These are some four classes of subclasses of IgG antibodies. Ig1, it is occurs available in excessive range, that is most abundant antibody, and it has a special character, that is it has a strong antigen binding feature or capacity. Then Ig2 mainly responds to the carbohydrate antigens, so these are the antigens which are found on the surface of the cells of the bacteria. So these carbohydrate antigens are found on the surface of the bacteria. For example, here is the bacteria. Okay, here is the bacteria, and we can see on the surface they are present. That is called as uh, carbohydrate antigens. That is the carbohydrate antigens. Here antigens means the foreign particle or foreign material which when enter into the body, that will triggers the immune system. Then next one is Ig3 antibodies. It is highly effective in complement activation. So Ig3 is act mainly by activating the complement system. So complement system means here some special proteins are going to involve. Which are going to work together to kill the pathogen. So for example, complement system means there are some group of proteins. Which are going to work together to destroy any pathogen. They are working together. Means they are the group of proteins. They are working in a group. That is why it is called as complement system. They are working in a group. That is called as a group of antigens. So this working of proteins in a group is called as complement system. So mainly this one, that is Ig3 antibodies. It mainly the mechanism of action is it mainly act through complement activation. Means Ig3 is going to trigger this one complement system. trigger this complement system. And in the complement system there are many proteins are there which are going to involve, and they are going to kill the pathogen. That is called as complement activation. The next one is Ig4 antibody. Ig4 antibody mainly plays a role in immune tolerance. Immune tolerance means here uh, it is the ability of the immune system to ignore the harmless substances uh, and prevent the attacks on the body's own cells. So Ig4 has a very important role in immune tolerance. Here, immune tolerance means, as I told you, it is the special character of the immune system which is going to stop the immune system in attacking the own cells, and also in attacking the harmless substances, so this is very important because whenever the immune system get hyperactive, it will not differentiate the harmful substance and the body's own cells. That is why Ig4 plays a very important role in maintaining the immune tolerance. These are the four classes of subclasses of the IgG antibodies, and their function. What are the specific functions of these subclasses? This is all about IgG antibodies. Structure of IgG antibody. So anyhow, we have already discussed regarding the basic structure of antibodies. So structure you can see here, it looks like here. Same, it has the two heavy chains, that is called as H chain, and it has two light chain, that is called as L chain. So these are the light chains, and these lengthy uh, chains are called as heavy chains. These are the heavy chains, and here we can see they are linked to each other by a disulfide bonds. Here we can see there are disulfide bonds. They are linked to each other by disulfide bonds. So it gives the strength, or you can say it will establish a link between the these chains, that is heavy and light chains. Then it looks like Y shape in structure. It looks like Y shaped structure with the two antigen binding sites. Here we can see there are two antigen binding sites. Here is the one antigen binding site, here is another antigen binding site. There are two antigen binding sites, and each IgG molecule has two regions. That is, one is constant C region and a variable V region. So here variable region, that is variable V region, is used for recognition of antigens. So it is responsible for recognition of antigen. Then that is another region, that is constant region, C region. So it is used to determine the function and interaction with the immune cells. So this constant C region will determine the how it is going to function, and how it will the or what is the interaction, how is the interaction with the immune cells. So this is what about the basic structure of IgG antibodies or immunoglobulins. Now the functions of IgG antibodies. IgG antibodies has their its own specific functions, which are going to discuss in this slide. The first one is stopping the infection. Here it will the IgG antibodies stop the infection by blocking the virus, bacteria, and other toxins, and preventing them from harming the uh, normal cells. So it plays a vital role in preventing the infection. That is, it will prevent the entry of virus or bacteria. Then marks the germs for destruction. So it will stick to the harmful microbes and make them easier for immune cells to recognize and destroy. So IgG antibodies will has a ability to stick on the microbes. For example, here is the microbe, and IgG antibody has a ability to stick on these microbes, and the it is very easy for the immune cell to detect the microbe. It will make the task easy for the immune cells. That is, it is going to bind with the microbes. Suppose this is the microbe, or any pathogen, or virus, anything is there. That is, it can easily bind with the microbes. Then that is marking the germs. It has a capacity to mark the germs, and make it easily recognizable for the immune cells, and helps in destruction of that harmful microbes or pathogen. Then it also activates the defense system, like it activates the complement system. So in the earlier slide we have already discussed regarding the complement system. That is, it is a group work. It is a group of proteins which are going to work together to destroy the pathogen or microorganism. So that is called as complement system. Then, so it will helps in stopping the infection. It will mark the germs for destruction, then activates the defense system, that is the complement system. Then it also has some other functions, like it helps helper killer cells. So IgG gives signals for natural killer cells. IgG gives signals for the natural killer cells, that is called as NK cells, to attack the infected or abnormal cells. So that is IgG antibodies will give the signal for the NK cells. Here we can see, suppose that is, here is the IgG antibody. Here is the IgG antibody, and here is the microbe, and here is the immune cell. Here is the microbe or pathogen. Here is the IgG antibody. So it will provide a signal to recognize or identify the pathogen or microorganism by the immune cells, so that it can helps in destroying the pathogen, and recognition of the pathogen by the immune cells. Then uh, next function is boost long-term immunity. So it provide memory for your immune system, ensuring the faster response when you face the same infection again. So boosting the long-term memory. For example, when our body get infected, or when our cell get infected by any pathogen, okay, after that is IgG antibody will helps the immune system or immune cells to recognize this pathogen, and it will destroy the pathogen by the immune system or immune cell get destroy the pathogen. Okay, so all these things are happening. Then along with this quality, it has another unique quality, that is called as a memory. That is called as memory. That is, it will boost the long term memory, means it will provide the memory for your immune system, ensuring a fast response. So whenever the same pathogen enter the body or enter the cell, it will easily recognize by the immune system, because it will provide a long term memory or immunity, so that the immune is aware of that old pathogen. So it will easily recognize whenever the same pathogen enter the body. The immune system get easily recognize the that old pathogen or the same pathogen, so that it will quickly provide the defense mechanism, or it can faster response and will fight against the infection, and the normal cells or body can be protected by the immune system. That is, it will not only helps the natural killer cells or immune cells to destroy the pathogen, but also it will provide a long term immunity, means by providing the memory for the immune system to destroy the same pathogen whenever it is going to re-enter the body. Then another function is it protects the baby. IgG has a capacity to cross the placenta. So placenta is a barrier which can be between the fetus and the mother. So IgG has a capacity to cross the placenta, give that is, and by crossing the placenta, it will protect the uh, fetus health. Fetus health. Uh, that is, it is providing or it is providing protection for the babies, that is fetus also. That is another quality of the IgG antibodies. And the last one is it has a capacity to regulate the immunity. Some IgG types helps in balancing the immune reactions, preventing the allergic and autoimmune diseases. See, whenever the immune system is get hyperactive, whenever the immune system is get hyperactive, so it will lose the recognition between the normal cell and affected cell or infected cell. So in those cases, the immune system will get produce a harmful effect on the normal cells also. So that can be avoided by the help of IgG antibodies. So whenever the immune system get hyper, it has a IgG antibody has a capacity to regulate the immunity and to balance the immune reactions, thereby preventing the autoimmune diseases and preventing the allergies. So that is the another function of that IgG antibodies.